diffusion demo of the Lab 1 diffusion and osmosis lab. And so first of all, to review, what is diffusion? Diffusion, remember, is the movement of molecules from an area of high concentration to low concentration. It's just due to molecular motion. So if we put some food coloring in water, food coloring is just going to move apart, and that's because the molecules are moving. And so it's going to move apart until the concentration throughout is going to be the same. And so basically what you're going to try to answer after watching this diffusion demo is rank the following from smallest to largest. Starch, glucose, water, the pores in the dialysis tubing, and then finally IKI or Lugol solution. So first of all, I should probably describe what all of these things are. Starch is going to be a polysaccharide, and so before we talk about what a polysaccharide is, we should talk about what a saccharide is, or a monosaccharide. Glucose is a monosaccharide. That means it's essentially C6H12O6, and so it is 6 carbons organized kind of in a hexagon. It's got a lot of oxygen and hydrogen around the outside, and so that's a monosaccharide. If we look at starch, so starch like that's found in a potato, starch is going to be a number of monosaccharides attached together, and so we call that a polysaccharide. And it's not just going to be a few like I'm drawing right here. A good starch or a good polysaccharide is going to have thousands of these glucose molecules attached together over and over and over. And so starch and glucose are both sugars, both carbohydrates. It's just that starch happens to be a little bit bigger. Hopefully you're familiar with water. Water is going to be made up of 2 things. You're going to have 1 oxygen atom, and then you're going to have 2 hydrogen atoms attached on the side. And so water is simply H2O. Next one is the pores in the dialysis tubing. So we're going to put some liquid inside of dialysis tubing. We'll seal it off on either side. So it's going to be sealed off like that. But the pores in the dialysis tubing are going to be these microscopic holes that are in the dialysis tubing. Remember, dialysis tubing is used in kidney dialysis. And so you can't really even see the holes at all, but they're really, really tiny. And then the last thing is Lugol solution, or IKI. The big thing that you're looking for here is going to be the iodine that's found in the Lugol solution. The big thing that you need to know is that if you ever have Lugol solution and starch in the same place at the same time, you're going to get a real blue color. So it turns to a blue color. In other words, if you were to take a potato and then just put Lugol solution on it, you're going to get blue coloration on that potato. That's just due to a chemical reaction, but we use Lugol solution, or IKI, as an indicator of starch, so where we're going to find starch. And so after you watch this demo, what you want to be able to do is rank the following: starch, glucose, water, pores in the dialysis tubing, and then finally IKI. You want to be able to rank all of those from the smallest to the biggest. Okay, so the video, since I'm at home, you can tell I don't have the chemistry equipment right here, and so I'm going to grab a video from YouTube. This one is I'm taking from Road to Utopia. It's got quite a few hits, 32,000. It's been around for a while, but it just shows you a demonstration of this. And so thank you to Road to Utopia. You probably want to visit it to see the whole video. I'm just going to show you a portion of it. And if you go there, you'll also see a really good video of a dancing robot toy, which is worth the trip as well. And so thank you for that. But this is the diffusion experiment. So first of all, let me kind of get it started. So let's get it started. Okay. So let me stop it right there and draw on it a little bit. So what do we have in this experiment? We have in here a bag. This is that bag of dialysis tubing. And you can see that it's kind of a whitish, kind of a yellowish color. It's kind of sealed off on the sides. And so if we look at what's in there, inside here, we're going to have 2 things. We're going to put starch in it to begin with, starch solution, and then we're also going to put glucose inside there. And then the other thing that we put, just to dissolve those, is going to be water. So we have those 3 things inside the dialysis tubing. Outside the dialysis tubing, so in this area out here, in the liquid portion of the beaker, we've got the, let me draw an arrow here. So out here, we've got water. And then we've also got the Lugol solution, so we have this IKI. Alright. The other thing you normally do before you start this is you start looking at the presence of glucose or the absence of glucose. And so you do that using these glucose test strips right over here. They're used to monitor glucose levels. So if you're a diabetic, it's a good way to see how much glucose there is in your urine is what we're looking at. Because if you have high glucose levels, it's an indicator of problems with regulating blood sugar. And so what you do is you put a strip into the fluid, and then it will change to a color. So it'll start as a blue color, but it can move all the way up to a red color. And this is what I use in class to figure out if there's glucose present. And so if we were to take this glucose strip and put it inside this, are we going to find glucose? The answer is yes. So we'll have a positive out here. And I just said over here a second ago that there's glucose in there, so that shouldn't be a surprise. If we were to take a test strip and look out here, we're going to find glucose out here. We'll have a negative for that. So there's no glucose out here to start with, but there's going to be glucose inside. And so this is what it looks like when it starts. So I'm going to have to get rid of this writing for a second, and then we'll play the video for a bit. And you can see immediately that it starts to get coloration change inside the beaker. And let me kind of move it up a little bit faster so we'll look all the way out here. And like play it there. So this is like 10 minutes later. And so now, 10 minutes later, when you pull it out, we got the same dialysis bag. It seems like the color has changed a little bit. If we were to now test it for glucose, what we would find is that there is still going to be a positive for glucose inside. But now if we look out here, there's going to be a positive for glucose out there with a test strip. And so that's kind of all the diffusion demonstration is. So now your goal is to figure out from smallest to largest, can you rank these 5 things? And you're going to have to use an understanding of diffusion, but you're also going to have to use logic to figure that out. And I'm not going to tell you the answer. So I hope that's somewhat helpful.
During the New York State Making Connections lab, you made the connection between pulse rate and exercise. So the first thing you had to do was you had to find your pulse. You take your pulse using the pulsation of an artery. Your artery is a blood vessel that is directly connected to the flow of blood out of the heart. And so you can actually feel the pulsating of the heart in the arteries. So you can either take your pulse by placing your fingers underneath your chin, or you can take your pulse at your wrist. You can't use your thumb to take your pulse because your thumb has an artery in it. So what you did was you took your pulse at rest. So your teacher either had you timed for 15 seconds, 20 seconds, 30 seconds, and during the time that was given to you, you counted how many beats or how many pulses you felt. So in this case, it looks like we are going to time you for 15 seconds, and we're going to multiply that by 4. The reason we're going to multiply it by 4 is because 15 goes into 60 4 times, and we all know that there are 60 seconds in a minute. If you took your pulse for 20 seconds, you would multiply your results by 3. If you took your pulse for 30 seconds, you would multiply your results by 2. So in this case, if we got results of 10, 12, and 10, we'd multiply that by 4, we'd get 40, 48, and 40 beats per minute. Again, we multiply by 4 because we only took our pulse for 15 seconds, and we want to multiply it by 4 in order to get our beats per minute. The next thing they're going to do is they're going to ask you to find the average. So what you do is there's 1, 2, 3 trials, so you're going to add them up, and I'm getting 128. Since there's 3 trials, I'm going to divide by 3, which gives me an average beat per minute, if I round up, of 43 beats per minute. Okay, so then we did this same experiment, but we exercised before, and we noticed that overall each classmate's pulse rate increased after exercise. And we made the connection that when you're exercising, you require more energy, and ATP equals energy. In order to make ATP, we need to go through cellular respiration. Cellular respiration requires oxygen. We collect oxygen in our lungs, but we need the oxygen to be at our muscle cells. So our circulatory system is going to have to push blood from our lungs to our heart to our muscle cells so that our muscle cells can use the oxygen to make ATP at our mitochondria through the process of cellular respiration. In the second part of the lab, we analyzed what happens if oxygen doesn't get to our muscle cells in time. So we analyzed 2 types of respiration, aerobic, which uses oxygen, and anaerobic, which does not use oxygen. So what we did was we took a clothespin, and for 1 minute you had to open and close that clothespin as many times as you can. So the muscle cells in your thumb were being worked. Okay, and what we noticed is over time we started to feel a cramping sensation in our thumb. And that's because at the beginning, oxygen was being delivered to our thumb muscles, allowing us to make enough energy through the process of aerobic respiration. But eventually, oxygen did not get to our muscle cells in time, but we were still demanding energy. Since we were still demanding energy, we needed to switch to anaerobic respiration. Anaerobic respiration does not use oxygen. It only makes 2 ATP and unfortunately produces lactic acid, which is what allowed us to feel that muscle fatigue, that cramping in our thumb. In the New York State Beaks of Finches lab, you guys pretended to be birds, and each bird had a variety of different beaks. So here are some samples that could have been your beak in class. You could have used a binder clip, chopsticks, clothespins, pliers, tweezers, or tongs. And what you did was first you competed for small seeds. So you were given a minute and you had to transfer as many seeds from the food source to your mouth one at a time. And you were timed. After the minute was up, you counted how many seeds you collected. If your average was above 13 seeds, you survived. You reproduced. However, if you were not able to collect an average of over 13 seeds, you had to migrate, and you migrated to the large seed islet. And again, you had to compete to see how many seeds you can get to the food source in 1 minute. If you could not eat an average of 13 small seeds, and you could not eat an average of 13 large seeds, unfortunately, natural selection took its course, and unfortunately, you died. So in this lab, you simulated natural selection. Organisms will fight for limited resources. Those that are best at fighting for that limited resource will survive long enough to reproduce. Those who can't fight for that limited resource have to find another limited resource. If they are not the best at eating another limited resource, they will continue to migrate. However, if they keep migrating and never find a food source, they will starve and will die and will not be able to reproduce, and they would be naturally selected out of the environment. So in this case, the limiting factor was the food, what they competed for. The environmental pressure they competed for was the food, and that's what picked and chose what organisms survive and what organisms die. The Peaks of Finches lab is represented sometimes on Regents exams by the wheel of finches. This is the wheel of finches. Notice we have a variety of different finches here on the Galapagos Islands. The way to interpret the wheel of finches is to pay attention to the center. So these organisms, which I'm using by green, all of these organisms are going to compete for mainly plant food. All of these organisms are going to compete for mainly animal food, and I highlighted the warbler finch because the warbler finch can only eat animal food. So the warbler finch does not have a chance to migrate. If the animal food is available, like the insects, it'll survive. If there's no animal food available, the warbler will die. So the warbler is the most vulnerable to natural selection because it only eats a very specific food source. So when you're looking at the wheel of finches, the first thing you need to know is who is going to compete with who. Organisms that eat the same food will compete with each other. So the vegetarian finch will compete with the large ground finch, the medium ground finch, the small ground finch, the sharp-billed finch, and the cactus finch. The warbler finch is going to compete with the woodpecker finch, the small tree finch, and the large tree finch. Additional information that you can find out is located in these circles. And this is just if the multiple choice question asks you about their type of beak. So you can use this chart. So for example, if we look at the vegetarian finch, it looks like its beak has biting tips. And if I look here, it has crushing bills. So if I were to describe the vegetarian finch, I would say that it has biting tips and crushing bills. So that's how you interpret the wheel of finches.
Biodiversity is a term that we use quite frequently when we talk about ecology. We remember that bio means life, and diverse means that we're going to have lots of different types of organisms. Thus, they would have lots of different types of jobs and roles in the community as well. The lab we're going to discuss today is one of our state labs that deals with relationships and biodiversity. The idea behind this is we were going to compare physical, chemical, and microscopic characteristics of a plant that was termed Botanicurus. And we are hoping to identify plants that are similar to this plant Botanicurus, because in the case of Botanicurus, it was described to us that this particular plant had a special chemical in it called Curol that was used in order to help treat people that had cancer. So there were a couple of things that we did. Our first test that we did was a physical type of test that dealt with structural characteristics. We compared the leaves and the stems and also compared the seeds of the plants. So I have two examples up here. I have Botanicurus, and I also have a species that was in term species Y. So Botanicurus, you can see, relatively small little plant. Some of you may even recognize that flower that maybe has been in your yard, kind of small looking little plant. You can notice that it has specific colored petals, specific numbers of petals and shapes. We also look at the stem and the leaf structure. Do the same thing for species Y. When you look at the seeds, you would look at the color of the seeds, the shape of the seeds. You would look at maybe any patterns or texture that you could see on them, relative numbers, and so on. The next thing that we did was we looked at them microscopically, and what we really wanted to look at was the stem structure and compare the stem structure of the different plants back to Botanicurus. What we were looking at when we were looking at the stem is we cut it as if you were to cut a loaf of bread, and we made prepared slides and looked at them under the microscope in order to look at what we call the vascular tissue. Vascular we know from our cardiovascular system in our body, which helps us to transport, In the case of plants, they have to transport materials as well. They transport food and water. In this case, they're termed the xylem and the phloem. The xylem is the tubes, the transport system that transports water, and the phloem transports the different food and nutrients around the plant. I usually try to remember phloem, starts with an F-type sound for food. So there's two different types of plants that we're looking for. One is termed a dicot, which has the vascular tissue bundled in a ring around the outer edge. And the other one would be a monocot, which has the vascular tissue very scattered throughout the entire structure. So we compared those between the different plants and Botanicurus. We then moved on to a little bit more of a higher tech kind of test, and instead of just looking at physical characteristics, we also were now going to look at some chemical characteristics. What we did was we did a paper chromatography, which chromo, you should think of color. Almost think of any time you hear, we talked about chromatin, we talked about those colored strands of DNA. In this case, we're separating out the pigments of the plants. We know that our plants have a pigment inside that helps with photosynthesis called chlorophyll, and they also have other pigments, which we happen to see in the fall. One of the things that we can do is we can compare the pigments that are hiding in the plants that maybe we don't necessarily see at this time of year, and compare those pigments to see if they have similar pigments from one plant to the next. So what we did was we took the plant, we crushed the leaves, and we ended up making an extract. That extract then was placed on a piece of paper, which was placed into a solvent, and then as the solvent gets absorbed into the paper, it draws those pigments on the paper and separates them out chemically so that you can see the other pigments that are being hidden behind the chlorophyll. And we compared it from plant to plant. Our next test that we did, we used an indicator that tested for a specific type of enzyme that we termed enzyme M. An indicator, remember, detects for the presence of a substance. We are indicating that it's present or not. If you remember, we've used some other indicators along the way throughout the year. We used a glucose indicator, which when we heated it, it was first blue. If glucose was present, it turned orange. We've used a starch indicator, which we typically call iodine, which started off as an amber or rust color and would turn a bluish black in the presence of starch. And then we've also used bromethymol blue, which was a test for acidity and carbon dioxide presence. It would go from blue to yellow if carbon dioxide was present or if it was an acidic solution. So this is a new indicator that we've done. We had this white powder that we mixed with the extracts, and when the white powder was mixed with the extracts, if it fizzed and bubbled, the enzyme was present. If it did not fizz and bubble, the enzyme was not present. Remember, an enzyme is also another term for an organic catalyst, which is used to speed up chemical reactions. They're made of proteins, and remember, our building blocks of proteins are amino acids. So if these organisms had similar enzymes, we know that enzymes are made of proteins. Proteins are directed for production through DNA. So if an organism has similar enzymes, then maybe it could suggest that they have similar DNA in that case as well. Moving on to something a little bit more technical, even further yet, we did some molecular tests, and in this case we tested the DNA through a process called gel electrophoresis. We've talked about this one a little bit before. Gel electrophoresis is used to separate DNA fragments based on size of the fragment. We use simple things called restriction enzymes. Once again, we're talking about enzymes. And the restriction enzymes cut the DNA in a specific sequence of bases. So it goes along and it uses special probes in order to highlight the DNA based on the sequences that are found. So in this case, if we were comparing, let's say, Botanicurus to species X, if you were high-tech enough to see the letters of the DNA-based sequences, you could see a sequence of, let's say, ATT, CCGG, and so on and so forth along down the road. Same thing with species X. When we cut the DNA with the restriction enzymes would cut in between the second C and the first G every time the sequence CCGG was found. So in this case, I would make a cut here, and I would make a cut there as well. We did this in paper to simulate it. This is done typically with a fancier setup than we used in our lab. So when I cut this segment up, I would end up with 1, 2, 3, 4, 5 base pairs for this particular fragment when it was cut into pieces. This next fragment would be 1, 2, 3, 4, 5, 6 base pairs long, and the last segment, actually, sorry about that, it would be 1, 2, 3, 4, 5, 6, 7, 8 base pairs long, and the last fragment you see here would be only 3 base pairs long. So when you get all of the segments cut up, you would take those fragments and you would place them into wells in a gel if you were to do this for real. Remember in the lab we did it as a simulation with paper. You would turn on the electricity, the DNA would run from one section of the well to the opposite end of the gel and separate it out based on fragment size, based on the number of bases. So 1, 2, 3, 4, 5 base pairs would end up as a dark line in the line where you would see 5 base pairs based on a standard. So in this case, we would have that fragment right there at 5 base pairs. In this case, this section of DNA was cut into 1, 2, 3 pieces right there. So you can see that we have Botanicurus and X, and we would compare the fragments. If they have a similar banding pattern, it would suggest that they have similar base sequences on their DNA in order to suggest that maybe they have a common ancestor. Remember that we can use this also for fraternity testing, crime scenes. We can do this for evolutionary relationships as we did in this lab. That we ran was an amino acid comparison, so getting even more technical yet. Remember, our amino acids make up proteins, so in this case we are comparing the building blocks of our proteins. And if you remember, protein synthesis is directed by your DNA. So if you have similar amino acid sequences, you would have similar proteins, which would suggest that you have similar strands of DNA. So if we started off, if you remember our protein synthesis, DNA gets transcribed in the nucleus into messenger RNA, which then leaves the nucleus to go out to the cytoplasm, to the site of the ribosomes, or it gets translated into amino acids using transfer RNA. In this case, if we see a DNA segment of TAC, GTT, ACA, the comparable messenger or complementary messenger RNA sequence would be AUG, CAA, UGU. That would then get translated based on our universal genetic code chart into a start codon, GLN for our amino acid, and our second amino acid would be cysteine. Remember, you look for the mRNA sequence in the chart, and it will tell you which amino acid would be brought in with that particular messenger RNA codon with an anticodon on the transfer RNA. Remember that proteins are polypeptides, meaning poly meaning many. So we have many amino acids put together in a chain, which we also call a polymer. So those proteins will help to dictate characteristics and traits. So if we have similar amino acid sequences and similar proteins, then maybe we know that we have those similar traits. So it could possibly be that the reason why those plants have similar colored flowers is because they had a similar DNA sequence that made a similar protein coloring that flower a specific color. So in the end with this lab, one of the things that we tried to look at was whether or not our DNA was similar between these organisms, and maybe that DNA was actually causing those similar characteristics. The more things that the organisms had similar to each other, the more it would suggest that maybe they had a common ancestor, and possibly these organisms would be a good match to look to see if they also made that particular chemical called curol.